Welcome to my YouTube channel. Please be sure to subscribe to my channel and also do not forget to press the bell icon so you never miss any video tutorial published by me. Hello students, welcome back to my YouTube channel Conquest Physiology. So today let us discuss one of the topics from Animal Physiology chapter from respiratory system. The topic is Oxygen-hemoglobin dissociation curve. So before we discuss about the curve, let us discuss some background information about it. Basically, oxygen in our body is transported in two ways. One of the ways is it can be transported in bound form. Bound form in the sense it is when it is bound to hemoglobin. It can be transferred in that way. In fact, 98.5% of oxygen is transported in this way. That means majority of oxygen is transported in bound form with hemoglobin. Secondly, it can also be transported in dissolved form, dissolved in plasma, and 1.5% is transported in this way. The reason why it is transported in dissolved form with plasma less, because oxygen is very less dissolved. Oxygen cannot dissolve in plasma or water very well. So that is the reason why it is not transported in dissolved form most of the time. Like very less amount of oxygen is transported in dissolved form, that is 1.5. Now let us discuss the structure of hemoglobin. This is like hemoglobin. It has 2 alpha chain, 1, 2, 3, 4. It has 4 polypeptide chains. And 2 of them are alpha subunits and 2 of them are beta subunits. Like alpha 2, we represent them as alpha 2 and beta. And these structures in green color, green dots, these are hemoglobin, heme, like iron part of hemoglobin. So basically it has 2 parts, polypeptide part like proteinaceous part and 4 heme iron molecules which combine with each other to form one hemoglobin molecule. Now the 4 iron molecules are in ferrous form. Like iron can be in Fe2 positive form. It is ferrous form, whereas Fe3 positive form is ferric form. So adult hemoglobin is in ferrous form, Fe2 positive form. So O2 binds with iron and the process is known as oxygenation. Right? Binding of oxygen with iron is oxygenation, and the binding is reversible because what is the purpose of oxygen? Why oxygen binds with the hemoglobin? The purpose is to transport oxygen to each cell of human body so that they can perform cellular respiration. Right? So once oxygen has already delivered, once hemoglobin has already delivered oxygen to the various tissues or cells of the body, now like delivery for delivery of oxygen, the binding must be reversible, so that like it's not like oxygen is binding with hemoglobin and it is bound to hemoglobin forever. That will be irreversible binding. But we need reversible binding so that once oxygen is delivered, like detached from hemoglobin in the body tissue, then the same hemoglobin molecule can be used again for the next time for oxygen transport. That is why it should be reversible. So it is reversible, and this is the reason why oxygen is easily delivered to the tissue. If binding will be irreversible, oxygen molecule will never leave the hemoglobin and it won't be delivered to the tissues. Simply speaking. Also, in fetus also there is hemoglobin, but that hemoglobin has 2 alpha and 2 gamma chains. The gamma chains have more affinity of with oxygen. That is why fetus, like small kids, they have very glowing skin. They have red, pink cheeks, red cheeks. So it's because of they have gamma. Also, one more thing we need to discuss is oxygen binding with hemoglobin shows cooperativity. Cooperativity in the sense, we know that there are four iron molecules in hemoglobin. So to each one of them, one O2 molecule can bind. One O2, second O2, then third O2, then fourth O2, right? Once one oxygen molecule binds to hemoglobin, then the binding of another O2 molecule is more easier than the first one, because this one bound O2 will help another O2 molecule to bind with hemoglobin. So this phenomena is known as cooperativity. And once second O2 has also bound to the hemoglobin, then third one will be even easier, because these two oxygen molecules will facilitate the easy binding of this oxygen molecule, and the fourth one will bound even more easily. So this phenomena is known as positive cooperativity. So this is the background information. Now let us discuss how exactly an oxygen hemoglobin dissociation curve or Oxy-hemoglobin curve looks like. Like it is sigmoidal in shape. Let us discuss that with the diagram. So now let us understand Oxy-hemoglobin dissociation curve. Like I already told you guys that the binding of oxygen to hemoglobin shows positive cooperativity. That means, like there are four sites of oxygen binding in one hemoglobin. So there are four like heme groups, right? And to each one of them, one oxygen molecule binds. So once one of the oxygen has already bound, then it helps the another oxygen molecule at second site to bind with hemoglobin. So it is known as positive cooperativity. Now let us understand this curve, Oxy-hemoglobin curve. That means percent saturation of hemoglobin with oxygen. The x-axis shows PO2, that means partial pressure of oxygen. So where exactly the partial pressure of oxygen will be higher? It will be it has units millimeter Hg, mmHg, like it is a unit of pressure, like bar is the unit of pressure. That is the unit mmHg. So the partial pressure of oxygen, which is indicated by PO2, is the most prevalent factor which determines how much O2 will be bound to how much O2 will bind to form Oxy-hemoglobin, right? So that means it means that more will be the PO2, the more will be the tendency of O2 to bind with Hb. That means in pulmonary capillaries, where PO2 is the highest, there oxygen will be bound to the hemoglobin with more affinity. But in body tissues where cellular respiration has occurred, so there is a lot of CO2, carbon dioxide. That means at those areas PCO2 is more. So that means in those areas binding the tendency of oxygen to bind with hemoglobin will be less. So that's how oxygen is delivered. Since PO2 is less in body tissues and PCO2, partial pressure of carbon dioxide, is higher. So that's why in body tissues the tendency of oxygen to bind with hemoglobin is less. So hemoglobin leaves the oxygen molecule, and that's how oxygen is delivered to the body tissues so that it could be used for cellular respiration. So this process is known as unloading of oxygen. This is how it works. So there are three states of hemoglobin in the body. First state is fully reduced. Fully reduced hemoglobin is reduction means loss of oxygen, right? So fully reduced means when there is not even a single molecule of oxygen bound to hemoglobin. That time it is known as fully reduced. So oxygen is not bound to any of the heme group of the hemoglobin. Fully saturated with O2, that means fully oxidized. Like when oxygen is bound to all the heme groups, all the 4 heme groups of hemoglobin, at that time the hemoglobin is known as fully saturated. Now the third thing is partially saturated. It means that like at some of the heme groups of hemoglobin, oxygen is bound. At that time it is known as partially saturated with oxygen. Now based on this, let us discuss what is the P50 value. The P50 value is PO2 of like partial pressure of oxygen at which hemoglobin is half saturated with oxygen. That means out of the 4 heme groups, 2 are occupied by O2 and 2 are not bound with O2. In our body it happens as 27 mmHg. Here I have indicated. Oxyhemoglobin, it is 50% saturated. It is known as P50. It is 50% saturated with oxygen when partial pressure of oxygen is 27 mmHg. Right? So I hope it is clear what is P50 value. Similarly, if 3 sites of hemoglobin in 3 heme groups of hemoglobin are saturated with oxygen, like if 3 O2 molecules are bound and 1 is not bound, that time it is 75% saturated, because like 3 by 4 into 100 will be 75%. Similarly, when only one oxygen binding site of hemoglobin is bound with oxygen at that time and 3 are not bound with hemoglobin, with heme group of hemoglobin, that time it is 25% saturated because one site out of the 4 is occupied by oxygen. So the saturation will be 25%. In the same way, when 2 out of the 4 oxygen binding sites are saturated with, like if they have O2 bound with them, then the saturation will be 50%, and this is known as P50 value. So I hope it is very clear that what exactly P50 value is. Now that was about the most prevalent factor, PO2, which decides how much oxygen will be bound to the hemoglobin at one point of time. But this is not the only factor which decides it. This is the most prevalent, no doubt, but other factors also decide. Like as I told you earlier, that CO2 also decides. If partial pressure of CO2 is more, then oxygen will bind less with less affinity with hemoglobin. That is how it is delivered to the tissues. Apart from this, there are 3 more factors, like apart from PO2, there are 4 more factors, like concentration of carbon dioxide, concentration of H plus ions, BPG. So there are 4 factors. Let us discuss those factors and how exactly they change this oxyhemoglobin curve in the body. Let us discuss those factors now. Students, these are the 4 other factors apart from PO2 which affect the binding of oxygen with hemoglobin, and they are the factors which can shift this black colored line, which shows the ideal condition of oxyhemoglobin curve. These factors can either shift this curve to left or to the right, depending upon how they are interacting, depending upon their concentration. Right. So the four other factors are H plus ion concentration, like proton concentration, which is pH. Carbon dioxide is second factor, which I discussed somewhat in earlier also. Temperature and formation of 2,3-bis phosphoglycerate, which is we use BPG for it, the symbol BPG for it. So let us discuss rightward shift first. The rightward shift in oxy-hemoglobin curve indicates decreased affinity of hemoglobin with oxygen. Due to this, P50 is increased. Like this is the rightward shift in red color. At this point of time, the P50 is increased. Like earlier, P50 was at 27 mmHg. Now it will be somewhere here, like around more than 30 mmHg. So it has, like, P50 value has increased over here. Earlier it was at 27 mmHg, but now it will increase. It will go beyond 30 mmHg even, right? So due to this decreased affinity of oxygen with hemoglobin, the unloading of oxygen from arterial blood to tissues will occur. It happens in our body tissues. This is the example. So it is very important. The decrement in the affinity of oxygen with hemoglobin is very important phenomena, because this way only oxygen which is bound to hemoglobin will ultimately get delivered to the tissues, and then it can be utilized for cellular respiration for ATP production. So rightward shift occurs when H plus ions like protons increase, and we know that increase in H plus ion concentration means decrease in pH. Increased carbon dioxide, it is also associated with decrease with pH. We will discuss all these factors and how they affect oxyhemoglobin curve in very much detail. Right now we are just discussing it superficially. Increased temperature also affects binding of oxygen with hemoglobin, and increased 2,3-bis phosphoglycerate. That means 2,3-BPG. These are the factors which cause rightward shift in the oxyhemoglobin dissociation curve. Now let us talk about leftward shift in oxyhemoglobin curve. It indicates increased affinity of O2 with hemoglobin. So P50 value decreases, like it is here. This blue colored line is showing leftward shift in the oxyhemoglobin curve. P50 decreases, so O2 unloading becomes very difficult. At this point of time, oxygen is very well bound to the hemoglobin, and unbinding of oxygen from hemoglobin is extremely difficult. So if we will check it over here, it will be somewhere here, like at around 25 mmHg. But that means value of P50 has decreased. Like earlier it was 27 mmHg. Now it will be around 25 or 23 mmHg. So value has decreased. So at this point of time, O2 will not leave the hemoglobin, and it will be bound very well with the hemoglobin. So let us see now why leftward shift occurs. So a leftward shift in oxyhemoglobin occurs when H plus ion concentration decreases, CO2 carbon dioxide decreases, like in pulmonary capillaries there is a lot of oxygen and very less carbon dioxide. Decreased temperature and decreased 2,3 BPG. So let us discuss these factors in detail. So students, now let us all the 4 factors which affect oxyhemoglobin dissociation curve in detail. So first factor is H plus ion concentration, which is known as acidity also, or pH. Increased acidity means decreased pH. As acidity increases, we know that our cells, body cells, are metabolically very active and they keep on producing 2 types of acids, lactic acid and carbonic acid. So as acidity increases, affinity, increased acidity means decreased pH. When H plus ion concentration increase, then it decreases the pH, because pH is negative log of H plus ions. pH is negative log of H plus ions. So when H plus ions in the body will increase, that time pH will decrease and acidity will increase. So when H plus ions will increase, binding affinity of hemoglobin with oxygen will decrease. So oxygen will tend to leave hemoglobin and diffuse into the body tissues. This phenomena is also known as Bohr's effect. So decreased affinity of O2 will lead to unloading of O2 from arterial bed to the body tissue. But we know that hemoglobin has buffering capacity. Like if H plus ions are increasing in the body, then hemoglobin can buffer them. But the thing is, when these H plus like protons will bind with the amino, we know that hemoglobin has a polypeptide part and also heme part. So polypeptide part is made up of amino acids. So when these H plus bind to the amino acids of hemoglobin, at that time they alter this structure. They alter the structure of hemoglobin in such a way, that oxygen carrying capacity of hemoglobin is decreased. So that's how an increase in H plus ion concentration in body will cause decrease in the affinity of hemoglobin with oxygen. So the curve will shift to rightward. Now the second factor is partial pressure of carbon dioxide. It also makes the same kind of effect, because it is partial pressure of carbon dioxide is related with like acidity in the body. Let us discuss how. PCO2 and H plus are related factors because low pH results from high PCO2. Like when partial pressure of carbon dioxide is high, that time H plus ion increase, and increase in H plus ions means decrease in pH. Let us discuss how they do. This is one equation. When CO2 enters our blood, at that time it binds with water present in the blood, because we know that our blood has some liquid part also, which is made up of water. So they both combine to form carbonic acid. This is carbonic acid. This reaction is catalyzed by an enzyme known as carbonic anhydrase. So carbonic acid is increased, and this carbonic acid has a tendency to dissociate in H plus and bicarbonate ions. So when it will definitely dissociate into H plus, and that means it will increase H plus ion concentration. And once An increase in H plus ion concentration will create the same kind of scenario, the same kind of condition which condition 1 is creating. H plus are increasing in the body, so the condition will be the same as first condition, like H plus will increase and they will bind to the structure of the amino acids of the hemoglobin, and which will alter the structure of hemoglobin, and they will decrease the affinity of hemoglobin with oxygen. So the oxygen, as a result of which the oxygen will be the unloading of oxygen will be facilitated from arterial blood to body tissues. So these are the two factors. Let us discuss other factors now. So the third factor which affects binding of hemoglobin with oxygen is temperature. An increase in temperature will shift the oxyhemoglobin curve towards right. We know that oxyhemoglobin is what shape? It is sigmoidal shape. So the increased temperature decreases the affinity of hemoglobin with oxygen and leads to unloading of O2 into the body tissues. So that is the same thing. As temperature will increase, O2 and unloading from hemoglobin will occur to the body tissues. So we know that the cells which are metabolically very active, they produce a lot of heat because they perform cellular respiration, and during cellular respiration heat is produced. Right, so metabolically it occurs in metabolically active cells, and in those cells unloading of oxygen from arterial blood to the body tissues occurs. Now the fourth factor is 2,3-bisphosphoglycerate, that is BPG. Earlier it was known as diphosphoglycerate, DPG, but now we term it as 2,3-bisphosphoglycerate. So this is produced during glycolysis. BPG binds with two terminal amino groups of 2 beta chain. There are 4 chains, right? 2 alpha and 2 beta. So it binds to the 2 beta chain of hemoglobin. So HB binds with O2 with less affinity. Once BPG will bind to the hemoglobin, then obviously it won't let oxygen to bind, because the site will be occupied with the, like, not the heme site, but the thing is it will create the situation like the affinity of O2 will be decreased with HB. So that means so we know that our body tissues are performing cellular respiration, and glycolysis is a part of cellular respiration. So it occurs in metabolically active cells. So that's how it leads to the unloading of oxygen from arterial blood to the body tissues. And one more thing I would like to cite, that people living in high altitudes naturally have more BPG. Apart from this, some hormones like epinephrine and human growth hormones, etc., they also cause production of large amount of BPG. So these are the 4 factors other than PO2 which affect the binding of hemoglobin with